On a full moon night in June, on a dark beach in Delaware Bay, the tide comes in and the beach disappears. Not underwater, under horseshoe crabs. Hundreds of thousands of them, hauling themselves out of the surf in the dark, their domed brown shells catching the moonlight, their long spiked tails dragging furrows in the wet sand. The females dig into the beach and lay eggs by the thousands. The males cluster around them, sometimes 4 or 5 deep. The sand between the tide lines turns bronze and brown and moving. A living carpet of ancient animals arriving to do what they have come here to do for longer than we can easily imagine. We call them horseshoe crabs. We think we know what they are. A living fossil, a relic, an animal so unchanged across geological time that a photograph of one beside a 450-million-year-old fossil looks like a matching set. Ancient, primitive, frozen. Every piece of that story is wrong. Not about the age. The horseshoe crab lineage is genuinely one of the oldest in the animal kingdom, and its persistence is one of the great unexplained facts of evolutionary biology. But frozen it is not. Primitive it is not. And the story of where horseshoe crabs actually came from, how they relate to the rest of the animal kingdom, what the genome says about the so-called living fossil, and what we have quietly done to an animal that outlasted the dinosaurs by 400 million years. That story has been almost completely rewritten by ancient DNA, and the rewriting contains at least one revelation that should have changed everything the moment it was published. This is what the DNA actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The textbook story of the horseshoe crab was a story told entirely through shape. Look at a horseshoe crab and it looks like nothing alive today. The domed carapace, the compound eyes on the sides, the long rigid tail spike called a telson, the book gills fanning on the underside, the 5 pairs of walking legs hidden beneath the shell. It is a body plan that strikes most people who encounter it for the first time as genuinely alien, and yet the fossils match. The Limulus polyphemus crawling up Delaware Bay tonight is, in its body architecture, extraordinarily similar to Mesolimulus walkii, a horseshoe crab fossil from the Jurassic Solnhofen limestone of Germany dating to 150 million years ago. Push further back to Paleolimulus from the Permian, and you still see the same essential form. Extend the comparison all the way to the Ordovician, to Lunatospis aurora dating to roughly 450 million years ago, and the shape persists. The same dome, the same tail, the same general plan. For most of the 20th century, this pattern led to a single conclusion. The horseshoe crab had stopped evolving. It had found a body plan so effective that natural selection had nothing left to improve. It was the textbook definition of a living fossil, a phrase Charles Darwin himself had coined, and the horseshoe crab was one of the most cited examples on the list. There was also a confident claim about its place in the animal kingdom. Horseshoe crabs, despite their name, are not crabs at all. They are not crustaceans. The textbooks correctly identified them as chelicerates, placing them in a group that includes spiders, scorpions, mites, and ticks. And within the chelicerates, the standard story placed them as the sister group to arachnids, close relatives but a separate lineage, having diverged from the ancestors of land-dwelling spiders and scorpions sometime in the Cambrian or Ordovician, then going their own way into the sea while their relatives conquered the land. Clean, logical, ancient, stable, the genome disagreed with almost all of it. The first major challenge came not from a single study, but from a cascade of phylogenomic analyses published through the 2010s and into the 2020s, each analyzing larger and larger gene datasets across broader and broader samples of chelicerate and arthropod diversity. The question these studies were designed to answer was precise. Where exactly do horseshoe crabs sit within the arachnid tree? The answer, when it arrived, was arresting. Horseshoe crabs are not the sister group to arachnids. They are nested within them. The genomic data, confirmed across multiple independent analyses using different gene sets and different computational methods, place Limulus and its 3 living horseshoe crab relatives, the 4 species of Schiphosura, inside the arachnid lineage. making horseshoe crabs not the closest relatives of spiders and scorpions, but something closer to their cousins, embedded within a group that everyone had previously assumed was land-dwelling only. This single finding demolished the clean division between marine horseshoe crabs and terrestrial arachnids that the textbooks had drawn. The division is not as clean as it appeared. The horseshoe crab is not on one side of a fork with all arachnids on the other. It is on a branch that grows out from within the arachnid tree itself, which means the common ancestor of horseshoe crabs and their closest relatives was probably already well along the path toward the body plan that eventually produced scorpions, spiders, and the rest of the arachnid world. Whether the horseshoe crab ancestor came from the land and returned to the sea, or whether its lineage diverged from terrestrial-bound relatives before the conquest of land was complete, remains one of the genuinely open questions the genome raised rather than answered. But the question itself is new. It was not a question anyone was asking as long as horseshoe crabs were safely placed beside the arachnid tree instead of inside it. The second revelation from the genome concerned the living fossil question directly, and it is the one that most completely dismantled the received story. When researchers sequenced the genome of Limulus polyphemus and compared it at the molecular level to other chelicerates, they did not find a genome that looked frozen. They found a genome that had been busy. The horseshoe crab genome is enormous. At roughly 1.7 billion base pairs, it is somewhat smaller than the human genome, but its internal structure told a different story than its external shape suggested. The genome showed extensive duplication events, regions where large segments of DNA had been copied and retained across evolutionary time, producing expanded gene families that had then diversified within each copy. These are the signatures of active molecular evolution, of a genome exploring new functional territory, not of one that had stalled. More specifically, the Hox genes, the master regulators of body plan development that specify what structure grows where along the body axis, showed something unexpected. Horseshoe crabs have undergone ancient whole genome duplication events, a rarer and more dramatic form of the same process. When a whole genome duplicates, every gene suddenly exists in two copies, freeing one copy to mutate and potentially acquire new functions while the other maintains the essential original role. Whole genome duplication is one of the most powerful drivers of evolutionary innovation in animal history. The fact that horseshoe crabs have experienced it means their molecular history is one of expansion and diversification, not stasis. The body stayed the same. The genome did not. The living fossil, it turned out, had been quietly innovating at the molecular level for the entire time its shell was sitting still. The third revelation from the genome is the one with the most immediate consequence for the world outside research papers. For decades, the horseshoe crab has been harvested for a substance in its blood. Horseshoe crab blood is pale blue, colored by hemocyanin, a copper-based oxygen carrier instead of the iron-based hemoglobin that makes our blood red. But the color is not why the pharmaceutical industry wants it. What the industry wants is a component found in the blood of horseshoe crabs called Limulus amoebocyte lysate, or LAO. LAO is one of the most sensitive detectors of bacterial endotoxins ever discovered. Endotoxins are the toxic compounds released by certain bacteria, and even in tiny concentrations, they can cause life-threatening immune reactions in humans. Every injectable medication, every vaccine, every intravenous fluid, every medical device that enters the human body must be tested for endotoxin contamination before it can be used. For decades, LAO has been the gold standard test for this contamination, an extract from the blood of a marine arthropod that has existed since before the dinosaurs has been the gatekeeper of pharmaceutical safety for the modern world. The genomic studies changed how researchers understood why LAO works the way it does. The amoebocyte cells that produce it are an ancient immune system component, a first responder defense against bacterial infection, whose molecular architecture the genome revealed to be extraordinarily conserved, maintained with remarkable fidelity across hundreds of millions of years of molecular evolution. The very features of the horseshoe crab immune system that make Lao so sensitive to endotoxins are the features that have been under intense purified selection for the entirety of the animal's evolutionary history. The genome showed that while the body plan was holding still and the internal genome was diversifying, the immune genes were doing something different still. They were being refined to a precision that nothing else on Earth has matched. The horseshoe crab's blood is not a primitive holdover. It is a highly optimized system that 450 million years of bacterial challenges have pushed to a state of sensitivity that human biotechnology, despite decades of trying, has only recently been to approach. The pharmaceutical industry harvests roughly 600,000 horseshoe crabs per year from the American Atlantic coast for their blood. The animals are bled and returned to the water. Between 10 and 30% of them die. The ones that survive often show behavioral and reproductive effects for weeks after the procedure. The females, who must store energy to spawn, may be particularly affected, and the population is in trouble. Delaware Bay, which hosts the largest horseshoe crab spawning aggregation on Earth, has seen its spawning population decline dramatically since the 1990s. The eggs those crabs lay on Delaware Bay beaches are not just a reproductive output. They are a critical food source for migratory shorebirds, particularly the red knot, a bird that flies from the tip of South America to the Canadian Arctic every spring and depends on the calorie-dense horseshoe crab eggs to fuel that journey. When horseshoe crab egg densities fall, red knot body weights fall, and survival rates fall with them. The two species are locked in a relationship that the horseshoe crab has sustained for millions of years, and that a few decades of over-harvesting has pushed toward collapse. The fourth horseshoe crab species, the ones that live in Asian waters rather than the American Atlantic, have their own crisis. Tachypleus stridentatus, the tri-spine horseshoe crab of the Indo-Pacific, is classified as endangered. Carcinoscorpius rotundicata and Tachypleus gigas, found across Southeast Asian coastlines, face similar pressures from coastal development, pollution, over-harvesting for food and traditional medicine, and the destruction of the sandy beach and mangrove habitats they depend on for spawning. The genomic work that established how closely related the four living species are to each other also made clear how little redundancy there is in the group. Four species, all under pressure, no close relatives that could pick up the ecological role if any were lost. The genome also added a layer to the ancient survival story that the morphological record had not been able to tell. When researchers looked at the rate of molecular change in horseshoe crab genomes across deep time, using multiple calibration points from the fossil record, they found something that helped explain the body plan stability. The horseshoe crab lineage has an unusually low rate of molecular evolution in its developmental genes, the genes that specify how the body is built. In most animal lineages, these genes are under moderate selective pressure, enough to allow the gradual accumulation of changes that shift body form over millions of years. In horseshoe crabs, those developmental genes appear to be under unusually strong purified selection, meaning mutations are being removed almost as fast as they appear. Because the existing body plan is so functionally effective that any deviation from it is costly, this is not stasis because nothing is happening. This is stasis because the environment is ruthlessly eliminating everything that tries to change. The horseshoe crab's body is stable for the same reason a perfectly engineered bridge stays stable, not because nothing is pushing on it, but because it has been built to absorb every push without bending. The genome showed this most clearly in the genes controlling the book-gill structure, those strange leaf-like respiratory organs on the underside of the crab that are evolutionarily ancestral to the book lungs of terrestrial arachnids. The book gills of limulus and the book lungs of spiders are homologous structures, different expressions of the same ancient design, and the genome now tells us they diverged from a common ancestral architecture that was already functional before the ancestor of either lineage had committed to sea or land. The horseshoe crab kept the original. The arachnids modified it for life on dry ground. Both modifications are still working 400 million years later. What does this mean for the creature hauling itself up Delaware Bay Beach on a full moon night? It means that animal is not what it looks like. It is not a relic. It is not a leftover. It is a highly optimized biological system whose external stability conceals an interior history of molecular expansion and immune refinement that nothing we have built can yet replace. It survived the Ordovician extinction and the Devonian extinction and the Great Permian Triassic catastrophe and the end Cretaceous impact. It did this not by being frozen, but by being precise, by holding its most important features stable while allowing itself to diversify internally in ways the shell does not reveal, and it is now being bled, harvested, displaced from its spawning beaches, and pushed toward population collapse in exchange for a compound that our pharmaceutical industry depends on, but is only now learning to synthesize, the synthetic alternative, a recombinant factor C test that can detect endotoxins without any horseshoe crab blood, exists. It has been validated. It works. The transition to it has been slower than it should be, held back by regulatory inertia and decades of institutional investment in the existing system, for 150 million years of survival through 5 mass extinctions. And the thing that may finally bring the horseshoe crab to the edge is a medical industry that needs its blood and has not yet finished choosing a different way. In Delaware Bay on a June full moon, the tide goes out slowly. The crabs that came up with the water begin their retreat, dragging themselves back toward the surf, many of them flipped by wave action, righting themselves or dying on the sand where the birds find them. The eggs they laid are already beginning to incubate in the wet sand. The red knots are already dropping out of the sky toward the beach. The ancient relay continues. It has continued through every catastrophe this planet has produced. The genome running inside that dome shell is not primitive. It is not frozen. It is one of the most refined biological instruments on Earth, built by 450 million years of pressure into something we barely know how to read and are only now beginning to understand, that we cannot afford to lose. The DNA is only beginning to tell us what it knows. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.